Thank you very much for the opportunity to come up today to Sydney. It's great to be back here. As you've heard, I've recently spent some time overseas. I've been in Scotland for the last 3 years. Really, my major focus there has been establishing the use of genomics within the NHS Scotland. So working out how you could use some of this sort of new technology I'm going to describe today to look at everything from rare genetic disease to improving the potential treatment of patients with cancer. And what I'm going to plan to do today, the title I've given you here is Personalised Medicine, a paradigm shift in oncology. I'd like to describe to you how we've seen just in the last couple of years a real push to move from when a patient presents with a cancer, we look at the organ where that cancer starts, and then we decide on how we go and treat that patient based on the experience of successes and failures on very, very large cohorts, where we look at overall benefit, where we look at the benefit of a population, not the benefit of an individual. And what we're seeing now, for the first time, is the ability to start to decode patients one at a time, understand the root causes of their cancer, understand why they actually had mutations in the first place, look for is there a specific Achilles heel that may be available in that patient, and then can we take that information and use it to individually decide what would be the best through to what would be the worst drugs to use in that patient. It's early days, but what you'll see is that there is a huge international effort to drive this forward, and we're looking forward to see how it will make some real changes in the next couple of years. Look, what I'm planning to do today, really, I want to highlight this idea of what I'm calling precision oncology or personalised medicine, and why is there a need. I'd like to then go on to what I call know your enemy. We're only just first really starting to get a good handle on why cancers start, and what's been the root causes of those cells going out of control. The next part is how do we, once we have all this sort of information, how can we try to come up with rules, or how can we find ways to shorten the odds of, or take some of the guesswork out of chemotherapy for patients when they come through the door. And then finally, I think you'll see that we're getting a lot of insights here, and while insights are great for boffins like me, ultimately this is all about improving outcomes. So we have to take this into the clinic, and we have to work at how we change from our current efforts into a more personalised approach, and that will take a united effort by everyone in this room going forward. So look, in terms of, that's going to be really frustrating. Could you please, is it that one?
Must be going 4 positions.
Oh, okay.
So we just
Perfect. That will work.
That should be okay.
That will be absolutely fab. All right, so traditional oncology and is really being based on the idea that you have a cohort of patients, and we will treat them with a series of drugs, and we know from those very large studies what percentage of patients will respond within that entire cohort, and based on those percentages you develop a hierarchy of the drugs that would be your first line therapy through to the drugs that you would use in second line, third, fourth, and that preordained list based on your very large population studies is the way you go forward and treat all the people thereafter coming in the lab. So you can imagine if this is a particular group, and if we, oh, an animation goes crazy. Let me try this one. Thank you. There we go. So if we have a particular group we treat with our first line therapy, there'll be a set of patients that respond to that out of our entire cohort that might come through for the year. Effectively, they are the people who will get a response. There will be a subset of patients that there has no effect on, and their disease progresses, and you may lose those patients. And indeed, there may be other patients that have adverse effects, which you lose from that cohort. And then what we would then see is after first line therapy, we look at second line therapy, same thing happens again. Third line therapy, same thing happens again. And then where possible, a fourth line therapy, and the same thing happens again. So we have this series of rounds of where we're treating our population based on our prior experiences of very large cohorts. That sort of approach has actually set us in pretty good stead. The data I'm showing you here now, I'm a scientist, so I have to show you some charts. I'll do my best not to get too scientific, but for a lot of cancers now, when we have a look at the 5-year survival, on average, we're seeing that those are really sitting around 50% for all cancers, and some we're doing remarkably well. But as you can see, there are some cancers there where, in fact, 5-year survival is very, very small. The red arrows here correspond to the cancers that I've spent the majority of my time recently working on, and indeed it's looking at a particularly nasty subtype of ovarian cancer, esophageal and pancreas, where we're finding that at the moment our traditional sort of approaches haven't worked. This next graph is a bit more challenging. This is showing you that over the last 40 years, how much that 5-year survival plot changed for each cancer. So for some cancers like prostate, we've seen really a dramatic increase in the 5-year survival over the last 40 years, whereas in the likes of pancreatic cancer, nothing has worked. Nothing has worked in the last 40 years. We've seen very small digit changes to 5-year survival stats. And so clearly the standard of care there needs to be rethought, and we need a new approach. I guess the other thing we've also seen with pancreatic cancer, and this is just re-highlighting that, is that this idea of taking a large population and then looking at trying to bring a new drug in and then trying to find the benefit, we're hitting an impasse. We're finding that you might get incremental gains, but overall, this is just showing the use of a new drug at the moment. You're talking about, when you go and do these studies on hundreds of patients, you've got a 7-week gain in survival overall for this particular drug. So we need to be thinking about maybe looking at a different way to combat cancer. The final thing I'll say about pancreatic cancer, it is one of those cancers that is actually on the increase. And I've taken the US data because it's the best model data at the moment. This year, more people will die of pancreatic cancer in the US than breast cancer. And indeed, it's expected to be more common cause of cancer death for it would be the second most common cause of cancer death by 2020, overtaking colon, and be well and truly placed by 2030 as the second most common cause. So for a cancer, in this case, this is a perfect example of where we might try to use a different paradigm. I guess the other thing I'm going to say is that some of the challenges I've highlighted here aren't unique to cancer. We know across the board the way that we use a lot of drugs to treat a variety of diseases. Many of the drugs we use don't work particularly well for an individual or get an overall benefit if we look at a large population, but there'll be substantial numbers of people that don't actually gain a response from our first-line therapies that we use at the moment. Cancer being one of the most challenging. And I guess the other thing that anyone who's experienced cancer or indeed knows people who have realises that we always have this challenge of adverse reactions, that the drugs that we use in our traditional approach have a wide variety of serious side effects, and indeed taking that stepwise approach from standard of care to second-line, third-line therapy, you're accumulating a lot of unnecessary damage. If we could only make the prediction of drugs more accurate and response in the patient in advance, you'd have a chance of actually saving a lot of difficulties and indeed saving a lot of money when it comes to the healthcare system. So I guess the idea of precision oncology, conceptually it's very, very simple. The idea is rather than going through in a stepwise hierarchical fashion that we've predetermined on big population studies, what we're going to do is take a cohort, we're going to decode and understand the root causes of every one of those tumours. We are then going to try to predict which, based on those mutations or those differences in each individual tumour, which ones are likely to be best matched to a particular drug. And in one assay, we should be able to take our cohort and divide them into those that will match our first, second, third, fourth line therapies. The patients that we don't think are going to respond to any of those drugs, we don't keep testing them with a lot of those other things unnecessarily. And then indeed, we then have to think about how we take our novel approaches and our new drugs and our new clinical trials to drive the cohort that we don't think we're going to have a good chance of helping with the standard approaches at this time. It's a very simple concept. The challenge to do so is absolutely enormous. I guess the ultimate goal of this idea of precision oncology is that if we take this approach, it should give, ultimately what we should be trying to do is improve the way we diagnose cancers. We're going to be looking at the mutations in the DNA to decide what type of cancer it is. We should be able to find a way to predict, we're going to need to be able to predict therapeutic response in advance, and that's how we stratify our patients. These same sorts of approaches, as you'll see as we go on, we should also be able to start to get better insights into what we call prognosis. So when a patient presents, if you understand that individual's cancer and you've got a large atlas of other patients and you know about their cancer experience, you have a better chance of actually working out whether this patient is likely to have a very aggressive disease or a better outcome. So prognosis is really how do we get a prediction of the path of that particular patient. And then finally, the idea that we should be able to improve safety. Don't use drugs unless we think that there's a good chance of them working, or let's try to minimise on those adverse effects as we have this genetic information to help us. Okay, so look, one of the things I've worked in pancreatic cancer since the 1990s. I won't say how early in the 1990s, but I started out looking at one gene at a time. What we've really seen recently is I guess we've always known for decades that cancer arises through the accumulation of damage to your DNA. We've never really been able to get a snapshot of all the damage you'll get to the DNA in a cancer cell, and if we were able to do that and capture it all and then systematically sift through it, we should be able to work out what's caused the cancer to grow in an individual, what causes particular cancer types to start in different organs. And I'm a bit of a military history buff, and I do like Sun Tzu's quote about in order to actually tackling something, you really need to understand the enemy, and once you do that, you really shouldn't, you've a much better chance of going forward. And in terms of cancer, if we really want to understand what it's doing and how to combat it, we need to understand why did it happen in the first place. We need to understand what things have gone wrong to make that cell grow and go out of control. There might be a whole pile of ways that a cell can go out of control, but ultimately when you're a patient, you want to know which ones of those events may give you, may act as an Achilles heel when it comes to a specific drug. So how can we sift down from all the damage that might be causing cells to go crazy to where's the vulnerability that we can get stuck into, and then finally let's think about how we take that forward to our patients. One of the things that's become very obvious to us over, I guess, the last 10 years with some of the technologies that are now being used is, and I've said to you that cancer evolves by accumulating damage to your DNA. Your DNA is like the hard drive in your cell. It effectively contains all the information that's encoded to make all the proteins in your body, and it's structured in such a way that it contains information so it can create tissues, it can do tissue repair, and we have all those magnificent processes and systems that we have within our entire body. And we now know that there's a whole variety of things that can go in and damage that hard drive. We can see exposure from the sun, so the likes of skin cancer here in Australia, that UV damage is actually going in and breaking our DNA and damaging specific bases. We have exposure to mutagens either in our environment from the likes of smoking or other sorts of carcinogens that we may be exposed to in our modern lives. We can actually have I should say, our body has a fantastic ability to repair damage to that hard drive. It can repair its DNA normally. Some people will inherit a defect in that, so they're predisposed to DNA damage, and that makes them predisposed to cancer development. Other people just pick up that damage by bad luck, and once you've lost your ability to repair DNA, you'll accumulate more mutations over time, and next thing you know, a cancer has a good chance of evolving. And then finally, pathogens with a Z. There's a mutation on my slide. That Z should not be there. A pathogen is something like a virus or a bacteria that we know in some instances can influence and promote a cancer to grow. And it's the accumulation of all of those things that ultimately could cause mutations over time. Those different mutations give the cells the ability to outsmart the immune system, to start to grow a bit quicker, to start to ignore some of the handbrakes that normally we see in cells, and ultimately, if you get enough mutations in the wrong number of genes, sooner or later you will see a tumour formed, and off it goes. Our challenge has been, we don't understand all the things that damage DNA. We don't understand how many genes can cause cancer when mutated. And we know that there's a whole pile of combinations of damage and combinations of genes that can give rise to cancer. But we don't know what is the total number of combinations. So this becomes a very big information problem, that we've got this thing that looks the same down the microscope, but at a molecular level or at your DNA level, we just don't know how complicated the story is going to be. We assumed it would be relatively straightforward. We now know that for many of those cancers where we don't see very good responses using traditional approaches, in fact it's very complicated at a molecular level, and we're going to need to factor that into our care going forward. So just to give you an example of what it looks like when you look down a microscope at the DNA within a cancer cell, this is just the way that we colour the chromosomes. So effectively each one of those colourful bands is a different chromosome. It contains that genetic information. You have 2 copies of each chromosome except for the X. So the sex chromosomes here, this is a boy. You can see an X and a Y down the bottom. We normally have 2 of each. Here's an example of what we would see if we go and look at a pancreatic cancer. Rather than having 2 of each cancer chromosome type, sometimes they're missing, sometimes we have 2, 3, 4, and the multiple colours show that the DNA has been broken and jammed and rearranged. Imagine if you took the hard drive in your computer and you started ripping pieces out and sticking it where it shouldn't go, taking one file and making 50 copies of it and sticking it everywhere else. You can imagine sooner or later you're going to get that blue screen of death on your computer. It's just going to sit there and hang. We've all had that when our computer's alive but it won't talk to us. That's effectively what's happening in cancer. The challenge for us is we don't know what is the specific damage that lead to that particular phenotype. In addition to this gross rearrangement and loss of control of our DNA, also if we went right down to the individual bit information on those chromosomes, this patient has about 25,000 other mutations where there's been little bits of damage, point mutations to the DNA. They've also got a germline mutation here which predisposes them to cancer. And ultimately, if I want to know my enemy, I need to decode all of that. And I need to actually sift out what were the drivers of this individual. So if we're looking at about 26,000 events, and there's really only 4 in this patient that are driving the disease, it becomes quite a challenge informatically to work out what that is. The wonderful news is that with the likes of big data and Google and all those approaches, if we collect enough patient data, we can find those needles in the haystack very, very quickly. I should say that the way that we can go forward and do that, it may seem like this problem is insurmountable, and I think really until about 2009 that it was, but we saw an advent of a new technology which allows us to decode your genetic blueprint, originally within months, now within days. When we decoded the first human genetic blueprint, it took about 13 years and about $2.5 billion US, about 1,000 PhDs working from around the world on it. When we sequenced the first Australian patients, looking at their normal DNA and then their cancer in 2010, there were 2 pancreatic cancer patients at that stage, cost us about $800,000 each, about 40 people working for about 7 months. I can say that the most recent patients we've done for a cost of about $3,000 to $4,000 and 4 people working on that, we can now do them within 5 days. And we're on track to be able to turn these around at prices similar to an MRI, I would suspect, by the end of next year, and we should be returning that data within days. So the technology is here. We are seeing a convergence of a lot of different skill sets coming together, but this type of Gordian knot, which is that mess of damage, we can now go through and decode in a computer using this thing known as next generation sequencing. I should say that when this work was done and first described, in fact coming from colleagues based in Cambridge, we realised, given how messy each genome would be, if we collected hundreds of patients from each cancer type and we stacked them all together, we should be able to find the genes that are being recurrently damaged, or which parts of your hard drive are being damaged recurrently, rather than what you just see by chance, by the random damage. And by looking for those signals, we'd start to find the needles in the haystack. And that approach was really only going to be possible if we looked at thousands of patients. So if we took 500 patients from each cancer type, say the 50 most common cancer types, that's 25,000 patients. We'd have to decode their genetic blueprint, define everything that went wrong in their cancer, and then start to look for signals. That's bigger than any one country, and there was an enormous effort called the International Cancer Genome Consortium, which was established to do just that. I was very lucky to run the Australian effort. We've worked on pancreatic, ovarian, esophageal cancers. In fact, we've also got another project which we've enabled now in melanoma. So we've been working through particular cancers and then comparing our experiences with groups in Canada, in Italy, in the US. And we've developed a way to share this information. It's an enormous amount of information per patient, but how do we codify it so that we can share this information and start to I guess, draw insights as we go forward in the future. I should say that this project is still running at the moment. It's due to finish at the end of next year. Our obligation was to sequence about 500 patients in total. I'm glad to say that we've actually ultimately characterised about 1,200 patients so far, and we continue to increase those numbers before the end of the program. The ideas are what I've spoken to you about here of decoding a patient, sharing that information, trying to work out where the key mutations might be in an individual. As I said when we first did it, took 7-8 months using a computer at the University of Queensland. It was a specially built computer. Cost about $4 million all up to build. Even then it was taking us too long. But the prospect of cloud computing, we hear about the cloud all the time now, has really allowed us to openly scale these sorts of approaches, whereby now we can analyse patients within days or hours. It's all just a matter of scale. So we've had to rethink the way that we do this. The nice thing about these approaches and the likes of ICGC is that when we've developed an approach here, it's been shared right across the globe. And in fact, we're seeing people using that internationally. So if that's sort of the atlas we're making and we're working out what's our Achilles heels, where do we go from there? Well, this idea of predicting the Achilles heels within an individual really relies on us doing a couple of things. The first thing we can do is just look at all the patients we've sequenced and say, are there any genes that we know when are damaged, they actually may make you sensitive or not to a particular drug? So we come up with hypotheses as to that mutation should correspond with that particular drug. And when we do that, we probably find that about 40 to 50% of pancreatic cancer patients have a potential vulnerability that we may be able to exploit. The challenge for us is that once you have a hypothesis, we need to get additional evidence to support it before we're really ready to take that into clinical care. A couple of ways that we've actually started to pull that together. One is that we've started with looking at our cohort and looking at how they responded to drugs which are given to them in the standard of care. Some of those patients really respond exceptionally well. And this is an example of a patient. The red line is showing you it's a measurement of a blood marker of tumour burden. This patient has had their tumour removed. It came back. They were given one round of a particular therapy, and then that cancer has effectively disappeared, both radiologically and detection in the blood. And when we've actually drilled into understanding what were the root causes of that cancer, we came up with an answer that we believe to explain that exceptional response. And if you use that as a hook to understand here's one that worked, we can then start to drill in and understand how we can exploit that in others. Or let's go looking for this particular pattern of damage. That crazy circle with those lines is basically a genome readout. But if we can find a patient with the same readout, we should be going forward and thinking about prioritising this approach. We can also do things where it's possible to take a piece of human tumour and grow it within a model system and then treat that with a variety of drugs. And effectively what this means is that this is just showing you the growth of a particular tumour over time. Gemcitabine is the standard drug that a patient would be given with one of these tumours. This is showing you that when it's got a particularly damaged genome, in this case it's losing a gene called BRCA2, it makes them exquisitely sensitive to a drug known as mitomycin C. So the standard of care, we're seeing that they're really having no effect on this patient, and indeed here we've got a drug that matches their vulnerability that works very well. Here's another one. Once again, it's a different gene being mutated. It's also causing vulnerability, and there's another DNA damaging agent which actually is able to, this the lines are actually showing you the rate at which the tumour grows. This tumour can't grow, whereas standard of care, it works absolutely fine. This is another way we build up an atlas of response and mutation, and we set that aside. And the idea is if we build these up enough and we collect these internationally, ultimately we should get to a point that when we get a readout on a patient, we should be able to put those two together. The final thing I'll touch on just in the last few minutes is this idea of transforming oncology one patient at a time. This might sound a bit like science fiction, decoding a patient, working out how you'll then take that into the clinic, doing it all quickly enough and robustly enough that we can try to make a difference. And then how do we actually start to assess that? This new model of understanding those defects and then taking that forward is one that is definitely gaining traction. And I should say that the idea of where we might see some benefit from these sorts of precision approaches, we can see that across a wide variety of cancers, there really does seem to be some potential benefit based on these global atlases that have been made on mutations. So how do we actually look to take that forward? Well, the first thing we need to do is come up with a roadmap of taking it forward. We need to understand the mutations. I've shown you we're putting all those together. We then need to get the rest of the system prepared to be able to use this information in a clinic. We need to be able to work out which drugs that are currently available should be used in a more clever fashion. We need to try to find other new drugs we could be actually targeting particular cancers. And then ultimately we need to get our entire healthcare system proving that there is benefit in this and then evolving our way to take it forward in the future. This sort of thing is not just science fiction. There are groups that are now working on it. In British Columbia, there is something known as the Personalised Oncogenomics Program. It's an NCI-listed cancer trial where they're personalising cancer approaches. Their goal is to do 5,000 patients. They've been running since the beginning of this year. And they're really working on patients that are at end stage and trying to find patients of last resort to see if now new personalised approaches where they will take a biopsy, sequence the genetic readout of the tumour, try to understand the consequences of that and create a report, give that to a group of clinicians, and then try to see if you can come up with a novel approach to treat that patient. It's very, very active. They have been in the media recently with some interesting results. If we scale up a little bit further, I've now hopped over to Germany. They have a personalised oncogenomics program. They've been scaling quicker than anyone else. They're up to their, they've already exceeded their 2,500 patients this year. And indeed, they're accelerating now to looking at doing every paediatric cancer patient in Germany using this sort of approach, whereby a patient will present, they will do this genomic analysis to understand the root causes in that individual. They'll get that to a clinic where they tie together where they think it may influence or change the treatment of that patient, and then try to get those patients into clinical trials wherever possible to prove the value of this approach in 2017. And it doesn't actually stop there. If you actually have seen this in the media, there are some very big efforts. France has just announced a staggering 670 million euros to transform cancer care, whereby looking at how do we take this molecular information and start to personalise cancer approaches. And they're looking that really between now and 2020, they want to do 235,000 patients. So they really want to test this in a real life setting. I think that number tells you two things: how challenging this is to transform the healthcare system and how big an ask it currently is to make this happen. I think that the French government is convinced this will give them benefit and save them money in the long run, and I think they really want to try to pioneer this and then be the leaders of transforming this medicine going forward. Finally, this isn't just limited to these national efforts. We can even see that our colleagues in pharma are now looking to do huge studies where they want to sequence patients in their millions. So if someone ever goes onto a clinical trial, even if it's not a precision approach, they want to collect that information and then start to bring precision onto their traditional studies. So the scale of doing 2 million patients with the amount of information involved, I think that's a biblical amount of information, but clearly, once again, with big data, these guys aren't scared of taking that forward. And I think we'll see some huge advances because of that in the very near future. Just finally, I wanted to say where I would like to see precision oncology going and what I think it will look like within the next 5 to 10 years. I think there are approaches here that we will see precision oncology creating something known as genomic pathology, and that is where we'll decode your tumour and try to come up with better descriptions around diagnosis and what else can we tell about that tumour. We'll move on to an area which I've called computational oncology, and that is a patient will present. You will actually compare that information to the experiences of 1 million other patients from around the world and the drugs that they were on and how they responded, and then you'll use that to start to build a model about how much you think about treating your particular patient. We'll also then see big advances in an area I call experimental therapeutics, and there is some really exciting work going on now where, similar to if you present with an infection, and the microbiology lab will take a sample of the bugs. They'll start giving you antibiotics, but then they'll go and work out the profile of every antibiotic and which one works on those bugs in your infection. Actually looking at doing similar model systems now where we'll take some of your cancer and within 3 to 4 weeks screen every drug that we have available in the laboratory on those cancer cells and come out with a sensitivity readout for the individual. That will really start to transform the way that we think about combinations, the way we think about giving an optimal drug dose to a patient, and may well help with the likes of safety and some of those things going forward. Ultimately, all of this will require a complete retraining from scientists through to our clinicians and allied health professionals, and then finally we ultimately still must prove that all this excitement that I have really does make a difference to patients at the end of the day, and indeed can we deliver healthcare in a more cost-effective fashion. So with that, I just want to list a small number of people that have really been driving this Australian effort. I have to say you that here it has been an enormous effort. It's been the largest project that's ever been funded by the NHMRC to date, but I'm hoping that these foundations will see some benefit in the very near future. Thank you.